Today I'm going to go over how we control the expression of genes. Why are certain proteins made when and why and all that stuff. That's what we're going through today. So keep in mind that gene expression does change over time. It can occur at any level, whether it's molecular, tissue, or organ, and sometimes it's a response to the environment. There's a common one in bacteria called the lac operon, and the lac is for lactose. So lactose comes in, that's the environmental response, and it tells this bacteria to make lactose or lactase, eat the lactose. Here's another example, though. We'll talk about globin. So, you got hemoglobin, and I mentioned in the last video how adult hemoglobin is made up of 4 polypeptide chains. There are 2 alpha units, 2 beta units. And which chain you actually make depends on how much oxygen level you're being exposed to. So as an embryo or a fetus, you will actually be making different hemoglobins than you do as an adult. So here's our typical adult hemoglobin. There's the 2 beta chains up here and the 2 alpha chains here. Each one contains a heme group, that's that blue diamond there, and that's what has the iron, and that's what helps to pull in the oxygen. And we find over time that where you are in the birth ways is different. So keep in mind, all throughout your life, you're making the alpha hemoglobin. That's pretty much standard. There's a couple of ones that are made really different, very, very young when you're an embryo that we're not going to go into, but they start very young and then they peter out as a fetus. So as a fetus, you're getting bigger and bigger. And as you're becoming a child, and basically you need more oxygen. The problem is where you're getting your oxygen. You're getting it from mom, and you're inside, and you need to get that oxygen from her. But you're not exposed to oxygen. You need to pull it from her. But her blood wants to keep the oxygen. So you need to have a type of protein that can pull that oxygen from her. That's why we have different types of fetal hemoglobin. So we'll have the 2 alphas here, but then we got the 2 gamma subunits. So those will be used instead up until you're born. So you can kind of see how the beta goes up, and then when you're born, the gamma goes down and beta goes up. And eventually you have all beta and very little gamma. The only time we actually see gamma after birth is usually in people with sickle cell anemia who have a genetic defect in that beta subunit. That's for another chapter. All right, so if we look at your blood plasma, so the yellow liquid that holds your red blood cells, it kind of feeds them and everything, there's 40,000 different types of protein. Not all at once. They change depending upon where you are in life. And we're looking at stem cell biology to kind of see how that's affecting it. How are certain genes turned on? When they're turned on? Why are they turned off? Because remember, every cell in your body has the same DNA, but they don't make all the same proteins. What is leading to that? Here's another great example. In your pancreas, you have exocrine and endocrine functions going on there. And that basically you have 2 different types of cells, exocrine cells and endocrine cells. They all start from the same stem cell. They'll then go down the endocrine pathway, their exocrine pathway. In fact, we know how to make cells go down the exocrine pathway. We expose them to transcription factor PDX1, and they will go down that exocrine pathway. So why is the pancreas important? Why do we need to know what pathway they'll go down? Well, if you notice, some of these things here may look familiar, glucagon, insulin. The other 2 may not be as much, but they're all relating to how sugar is absorbed into your cells. So basically, insulin's made by the beta endocrine cells. Glucagon's made by the alpha endocrine cells. Those are the two main ones, but some other people can have these delta causing problems too. So we know with the stem cell, once they go down the exocrine pathway, and we got this progenitor cell here, if it's exposed to PDX1, it's going to become a mature exocrine cell. We're not too concerned about that. We want these guys here because people who have diabetes have some issue here. So we want to go down the endocrine pathway, and then we want it to become either a beta cell, so it produces insulin, or an alpha cell that produces glucagon, or if you're missing one of these hormones, it'll go down that way. We haven't figured that out. So if you can figure that out, that's $1 million idea. You can give me a percentage for having you come up with that idea if you solve it. Another way we figure out about what kind of proteins are made is through a process called proteomics. This is a really up-and-coming field. You may want to look more into that later. But basically, the idea is that you're looking at what proteins are made over your lifespan and what environment you're in. So if we look at you as an embryo and you as an air-breathing human, you will see there are very different things being made here. The two biggest ones I'll discuss, transcription factors. Notice it's very little here, very big here. Transcription factors are factors that turned in a bunch of genes. When you're this kind of thing, you have a bunch of genes being turned on. There's that are going to do that. Once you're in a, air breathing and you're living on your own, your mom's not feeding you in the womb, you don't need as many transcription factors. That one break. However, we look at enzymes. Enzymes goes way up here once you're on your own. Why? Well, mom isn't digesting any food for you now, so you got to make all of those enzymes so you can digest your food. The embryo doesn't need all that stuff. So what turns on genes? What are the mechanisms involved? Well, there's a couple of things. One is the transcription factors. These are factors that actually turn on other genes, I mentioned before. There are sequences in front of the genes called promoters. Some of these promoters basically actively promote the making of that messenger RNA or making of that protein. So it puts this promoter on that RNA, messenger RNA, so it makes that protein. So it basically promotes these things being made. If mutations occur in those things, we can actually have less made or even more made, depending upon what goes on. Copies of the genes. There's some genes we just have more copies of, so we're going to make more of them. The histone code is also important. So histones, those are the proteins that your DNA is wrapped around, and depending upon how they're wrapped on that histone, they'll be displayed for easy transcription. And then adding various molecules to those genes can actually turn them on and turn them off, depending upon what's needed. So if we're going to turn it on, we would add acetyl groups or phosphate groups that would turn on transcription, or we could remove methyl groups, and that will turn it on as well. One way we turn off genes to make sure we don't make them is through making these RNA interference things. So these are messenger RNAs that will actually bind to this, it binds to a messenger RNA. So it's an interfering with a messenger RNA. So basically it silences the gene. It will not work. Another way is we just remove them. We remove it through that RNA interference. So once that messenger RNA binds to an interfering messenger RNA, we make double-stranded RNA. Well, double-stranded RNA just can't exist in our cell. So we actually have enzymes that are designed to break up double-sided RNA. The nuclease just shatters it, so it's no longer any good. All right. So ways we can turn off genes at the transcription stage would include chromatin remodeling. That would be with the histone code. RNA interference I talked about. Alternate splicing I'll mention here in a few minutes. Translation, protein folding that's folded incorrectly. We know it doesn't work. Polypeptides are shortened. They won't work then either. If it's broken down, we can add sugars, and I think we can do other things to turn it off at the translation stage. All right. One of the things I do want to point out, as I mentioned in the past, only of our whole genome code, only 1.5% of it actually codes for a protein. However, in that, we have 25,000 genes. From that 25,000 genes, we make 200,000 proteins. You've probably heard the story about one gene, one protein before. Well, clearly that doesn't take place. We got far many more proteins made with these genes than we thought. So two ways this can happen. That's through alternative splicing or several genes cut from a single gene. Alternative splicing, that's just basically the exons. When the introns are removed, the exons have to be put together. There's no reason why it has to go A, B, C, D. They can be rearranged, and we can get all types of different possible proteins from that. So we can get an AC or an ABCD, or we can get even a DCBA, which would be something different. So we can do all of those kind of alternative splicing, makes all sorts of different proteins. This one's referring to something being cut from a large protein. So the disorder you're seeing there is dentagenesis imperfecta. Basically, there's a protein that we cut two different proteins from, DPP and DSP. So basically, the DSPP gene is transcribed and then translated, and then there's an enzyme that will cut, fold it into proteins. And then these proteins will allow enamel to be added to teeth. And when that happens, you don't get this. This is no enamels. Keep in mind, most of our genes do not code for anything. So what are they? Most of it's viral DNA. Those viruses, we just can't get away from it. Some of it are the non-coding RNAs, like tRNAs, rRNAs, things like that. The introns, the promoters, other controls are all there as well. We'll talk about pseudogenes at a different time. The centromeres as well as the telomeres are all part of the chromosome. They are DNA, so they're part of that as well. The histones, they actually are codes in the DNA as well. And that basically ends it. Notice this is a very short chapter, even though it's a very important subject. The thing is, we don't know a lot of this stuff. This is where a lot of the cutting edge research is going in. If you really want to go into this kind of field, there's a lot of research to be done here. Go to the Google Forms, answer the questions, and that ends control of gene expression and genome architecture.